we start our reaction with about 150 milliliters of deionized water. We are going to add a little ammonia to the water to make sure that the pH is slightly basic. If the solution is acidic, it will prevent all of our solid from forming. So we add the ammonia, we stir it, and then we test to see whether it's basic with a piece of red litmus paper. The litmus paper turns blue. This tells us our solution is basic, and we should be ready to add our two reactants and start the reaction. As we add our reactants, we use a little deionized water to rinse out the solid to make sure all of the reactants get into the sample. We stir it to dissolve the first reactant. And then we take the second reactant, add it. When we do, notice immediately precipitate starts forming and the reaction gets extremely cloudy. Now the particles formed initially here are going to be very, very small. This will create a problem when we try to filter the solution to remove those particles and dry them and weigh them. So we turn on our hot plate and we are going to heat up the sample for about 15 minutes and do a process called digesting the precipitate. The digestion process slowly warms it up, particles dissolve and then reprecipitate and form larger particles than were present initially. This will minimize loss of particles when we filter later in the experiment. To make sure we do not evaporate too much of the water, we place a watch glass on the top of the beaker and we allow it to heat up. Over the next 15 minutes, we're going to occasionally stir it and we will let the process continue. Digesting for about 15 minutes. We will now remove it from the heat and allow it to cool slightly and allow the solid to settle. In order to get as much of the solid out of the beaker as possible in the filtration process, we are also going to heat up a small amount of water at this point, which will be used to rinse out the beaker during the filtration.